Good morning, one and all. I myself am Likit Saifani Chowdhury to studying BTech Biotechnology in CPIT. Today, I going to present my project part 2 final review. I done my project under the guidelines of Dr. V. Aruna Ma'am, Associate Professor, Department of Biotechnology. My main title of my project was Enhancement of Secondary Metabolites in Bacopa monnieri, a medicinal herb. These are the title of contents where I going to present today. My main introduction. Recently, I studied some recent studies. I came to know that Bacopa monnieri is ranked second among the most important Indian medicinal herbs in terms of medical relevance and commercial worth. It has numerous pharmacological properties like digestive, endocrine, anti-inflammatory, antibacterial, as well as antioxidant properties. Additionally, the herb has been used to treat neurological and neurophysiotherapeutic conditions. Bacopa monnieri plant growing originally showed seasonal fluctuations, and the plants gathered from October through December were found to have highest content out of all the seasons. Callus culture can be started from the parenchymatous tissues of shoots, roots, and other plant parts. Callus consists of undifferentiated mass of cells growing on a semi-solid media and sufficient supply of nutrients including growth hormones such as auxins, glycerins, cytokinins, and kinetoplastid, as well as a maintaining of a sterile environment are required for the preservation of callus culture. This is the Bacopa monnieri plant that I am growing in our college CBIT. In biotechnology, elicitation is commonly defined as the induction or enhancement of metabolite production brought only by the presence of very low quantities of elicitors. Elicitors are compounds that can stimulate the production of secondary metabolites in plants as well as the production of new secondary metabolites. According to research, Bacopa extract contains tannin, flavonoids, glycosides, terpenoids, saponins, and steroids as well as bacosides A, B, C in and but lacks anthocyanins, phenolic, etc. in callus, cell suspension, and elicitation culture. The preliminary phytochemical research received a great deal of attention, and the raw pharmaceuticals have a diversity of secondary metabolite compositions. Phytochemical extraction is the separation of the therapeutically useful portion of plant tissues using well-established methods and particular solvents. The initial plant material used the extraction solvent such as methanol, ethanol, chloroform, ether, or acetone, utilized to obtain secondary metabolites like tannin, terpenoids, steroids, and other compounds like bacoside A, B, and C. And the extraction procedure itself were the three main factors affecting an extract quality. In order to select which species to focus on for high performance liquid chromatography, that we calling it as a HPLC analysis, while studying a large quantities of secondary metabolites from plants, it is essential to have the tools necessary to characterize the sample. To do this, straightforward biological experiments can be used. A very versatile technique is HPLC to find measure, and purify each of the component parts. Analytical chemistry and phytochemistry both employ HPLC, a chromatographic technique that can discriminate between a number of chemicals. My main aim to enhance secondary metabolite production in Bacopa monnieri through callus culture and elicitation, usually secondary metabolites that are not directly involved for normal growth, development, and reproduction. My main objectives are optimization of media and establishment of callus culture in Bacopa monnieri. To establish callus culture in Bacopa monnieri. Next objective was to enhance secondary metabolites in Bacopa monnieri through polymeric nanoparticle elicitation. Next objective was extraction of phytochemicals for in vitro culture and ex vivo grown plants of Bacopa monnieri. And my next objective was HPLC analysis of bacosides for in vitro cultures and ex vivo as grown plants of Bacopa monnieri. My main novelty using polymeric nanoparticles as elicitors to enhance the secondary metabolites in Bacopa monnieri. These polymeric nanoparticles are absorbs loaded active substances into their polymeric core. This is my literature review. In this literature review, I came to know pharmaceutical important compounds of this plant as well as pharmaceutical applications of the plants, how it is being used in pharmaceutical, as well as how can increase the production of this plant through scale up technology. From this review paper, I came to know that how to improve plant secondary metabolites through biotic and abiotic elicitation, what type of techniques to be used, and how to increase their secondary metabolites in the plants. From this literature review, I came to know that how to enhance bacoside production. Bacoside is also a secondary metabolites of this particular plant, and by using suspension cultures as well as elicitation, and how to extract those cultures. Once we completed the extraction, how to perform HPLC and what type of peaks we're going to get in HPLC, I came to know from this particular literature review paper. These are my materials. My plant material Bacopa monnieri. My instruments are laminar airflow, pH meter, orbitary shaker, hot or oven, analytical balance, autoclave. My media was Mirage and Scoop media. My plant growth regulator hormones are 2,4-D and pictoram. 2,4-D is nothing but dichlorophenoxytic acid. My main chemicals are macro and micronutrients. My nanoparticle, polymeric nanoparticle. My first method was MS media callus induction with oxygen hormones. I had prepared MS media with different concentrations of 2,4-D and picloram in test tubes of zero point 0.1, zero point 0.5, 1.0, 2.0, 3.0 mg per liter. I had taken shoots with 4 to 5 nodes from healthy plants of Bacopa monnieri and I surface sterilized, and I inoculated the surface sterilized explant in MS media is supplemented with different concentrations of 2,4-D and picloram, and I incubated these test tubes in the light at 25 plus or minus 2 degrees centigrade until the explants induce callus from the cut ends. My second method was MS media callus induction with nanoparticle as well as oxime hormone. I had taken 1.5 gram of polymeric nanoparticle and I synthesized with grapefruit seed extract liquid with different concentrations of 0.5 ml, 0.75 ml, 1 ml, and I name it as a ABC, where A means 0.5 ml, B means 0.75 ml, C means 1 ml in 100 ml of distilled water, and I leave it for room temperature, and I perform mixing for 1 hour to form nanomycels, and then I characterize with SEM and TEM. Later, I prepared MS media with 2,4-D, zero point 0.2 mg per liter, as well as polymeric nanoparticles of A, B, C different concentrations like 0.5, 1, 1.5, to 2.5 ml. Later, I had collected 4 to 5 notes from shoots of healthy plants and I surface sterilized. And this surface sterilized plant I had inoculated in MS media is supplemented with 240 of 2.0 mg per liter as well as different concentrations of polymeric nanoparticles of A, B, C in test tubes. Then I incubated these test tubes in the light at 25 plus or minus 2 degrees centigrade until the explant induced callus from the curtains. This is my figure how I had inoculated this particular plant. My next methodology was phytochemical extraction. I had taken 5 grams of callus in 30 ml of methanol and 5 grams of callus in 30 ml of uh, methanol and 5 grams of callus in 30 ml of aqueous. In 3 solvents, I had taken 5 grams and 1 solvent in 30 ml. Later, later I lay at 100 degrees centigrade at water bath for 30 minutes. It means that I kept this three samples in 100 degrees centigrade water bath for 30 minutes. Later, I filtered this extract and I evaporated this extract to dryness. Once the dry extract is formed, then I store the dry extract till the time of study. We can study phytochemical screening as well as HPLC analysis, and I studied HPLC analysis. Compounds are separated by using HPLC based on their interactions with solid particles in a densely packed column and the solvent in the mobile phase. Before passing through the detector, the enlight must be eluted the column at high pressure of up to 400 bars. HPLC is useful for compounds that cannot evaporate or dissolve at high temperature. In a single process, HPLC may do both qualitative and quantitative analysis. I used Water Alliance HPLC system of 2695 model and PDA detector of 2996 model, and this HPLC analysis is used to determine Bacopa monnieri plant content in the phytochemical extracted sample of ethanol, methanol and aqueous water. Prior to injection in the HPLC system, these samples were diluted using mobile phase. This is the HPLC equipment, and this is my schematic overview of my work. My first step, I prepared the stock. Second step, I prepared my MS medium. Third step, I had transferred MS medium into test tubes. My fourth step, I had autoclave my MS medium which I had transferred in test tubes. Next step, I done surface sterilization. Later, inoculation followed by incubation and phytochemical extraction of the colors with the solvents, and later its last and final step is HPLC analysis. This is my result. Callus induction with different concentrations of oxygens and polymeric nanoparticle. This is my table. In this table tells about influence of various oxygens on callus induction from leaf explant of Bacopa monnieri after 4 weeks. For each treatment, 10 replicates were performed. The experiment was repeated twice. Number of plus signs represents the degree of callus, and I got black and brown compact callus, and this black and brown compact callus comes as nature of callus. And this table too completely tells about influence of oxygens and various polymeric nanoparticle concentration on callus induction from leaf explant. For each treatment, 10 replicates were performed and experiment was repeated twice. Number of plus signs represents the degree of callusing. This is the phytochemical extraction using ethanol, methanol and aqueous. This is the HPLC analysis of Bacopa monnieri were carried out for methanol, ethanol and aqueous extract of callus and plantlets. The chromatograms were collected and analyzed. Since Bacopa monnieri plant is freely soluble in ethanol, methanol aqueous. The callus and plantlettes were extracted in ethanol aqueous, the first run of blank to check the error from the mobile phase. This figures of uh, peaks of chromatograms. Usually, our standard is too. My standard is very costly, around 35 to 50,000, and sometimes it leads to a lakhs. So I had taken a review paper for reference to check the peaks, and I came to know from the review papers that this peaks comes as Bacosites A, B, C. So in my procedure of secondary metabolites, I came to know that Bacosites A, B, C of secondary metabolites are present from this particular peaks. That I came to know by taking analysis of research paper because my standards are very costly. And this is my discussion. Indian native herb Bacopa monnieri is also referred to as Brahmi. Madhyarasana has been used for million to boost mental and brain functioning and raise mental capacity. It is also used to treat diarrhea, water retention, asthma, and bronchitis. The callus that had formed at a base of nodal segments, polymeric nanoparticle elixires, were synthesized in the callus culture of the Bacopa monnieri plant to produce better phytochemical output. Phytochemical extraction is the isolating of bioactive compounds from plant material using different extraction techniques. These bioactive compounds are often used for medicinal, nutritional, or industrial purpose. The HPLC procedures are used to isolate and purify substances from plant-based sources for producing secondary metabolites. This is my conclusion. I can conclude that callus growth depends on concentrations of auxins and explants used, where enhancement of secondary metabolites in Bacopa monnieri plant through polymeric nanoparticle elixires, and it poses from phytochemical extraction for varied composition of secondary metabolites and for estimating quantity and qualitative of secondary metabolites by HPLC analysis. Where coming to my HPLC analysis, I came to estimate quantitative and qualitative analysis of secondary metabolites like Bacosites A, B, and C, where I observed the peaks from research papers and I came to conclusion that these three secondary metabolites are present. Usually my standards are too cost, where I kept a funding document where for BTech biotechnology no one, our college will not pay that much amount of funding. Only they can pay up to 10,000, not more than that. For that particular reason, I taken my research papers as base and I compared my peaks with research paper peaks with same same solvents and with same equipment. I came to know that these are the three metabolites are present like Bacosides A, B, and C. The future aspects of my project as promising future prospects of of Bacopa monnieri plant. These are ranging from development of new pharmaceutical products to environmental remediation and nutraceutical applications. Biotechnology can play a crucial role in the optimization of Bacopa monnieri cultivation, the development of novel products, and the exploitation of its therapeutic potential. Plant biotechnology offers several future prospects for the enhancement of secondary metabolites in Bacopa monnieri, including the development of novel varieties with enhanced profiles, increased yield, and improvement in quality. These advancements can lead to the concentration of secondary metabolites and their potential pharmacological applications in the treatment of various diseases such as Alzheimer's diseases, anxiety, depression, etc. My objective status: I had targeted attended all the objectives as per mentioned by my internal guide Dr. V. Aruna ma'am. These are my references where I had used in my project, and I used certain references And I want to acknowledge my guide, Dr. V. Arunam ma'am, and I want to tell big thanks and my sincere gratitude to her because of her. Today I can complete my project very successfully, and I love her guidelines. I can say later I want to appreciate my HOD, Dr. Ashtaush Pandey sir, and as well as I want to congratulate our principal, Dr. P. Ravinder Reddy sir, because of uh, because of them where I can able to complete my project, and they made my guide to they were allocated me an excellent guide. I want to thanks for them because of them only I got an excellent guide, Dr. V. Aruna ma'am. Later, my man make my HPLC analysis performance HPLC analysis performing in HVK Life Sciences with Dr. M. Navin Reddy sir. Where Navin Reddy sir is a director of SVK Life Sciences, I want to thank him for performing my HPLC analysis. And I performed my HPLC analysis with free of cost because of my guide Dr. V. Aruna ma'am. Later, I had done this HPLC analysis by taking the help of Dr. K. Dharmalingam sir. Later, I want to thank all my non-teaching staff for making me to do this project very successfully because they had made all the equipments ready for my projects. I want to thank all of them. My main project outcome, project paper manuscript were under preparations. And thank you.